Finish up specific immunity, we'll now look at cell-mediated immunity. So here in this diagram, it shows where everything begins with the immature lymphocyte. Will be a T lymphocyte. Well, it'll be a lymphocyte that will then migrate to the thymus as a T lymphocyte. And then at the thymus, it can become, depending on what genes it has, it can either become a CD4 cell or a CD8 cell. And you'll notice that a CD4 cell has a certain particular receptor type on the outside, and a CD8 will have a different receptor here on the outside. And these receptors not only are for the antigens, but they're also for the antigen presenting cells, whether they're class 1 MHC proteins or they're class 2 MHC proteins, which we'll get into in just a minute. So in the thymus is where either the CD4 or the CD8 cell will build its T cell receptor. So you can see these are two different cells. They have two different receptors on the outside. They will be activated by two different antigens. But both of them need to be activated by an antigen presenting cell. So in this particular case, once they leave the thymus and they go out into systemic circulation, whether it's the lymphatics or they're in the bloodstream, either a dendritic cell, which is the type of macrophage that's a resident in the skin, and so they will engulf, that macrophage will engulf the antigen that was found within the skin, that infected the skin, and then take it to typically the lymph node and present the antigen to the T cell. Now, in the case of CD8 cells, these cells are going to become cytotoxic T cells. And so let's go through what happens with them. So this is going to be an antigen that would be found in a body cell bearing a class 1 MHC protein. So the MHCs are basically like antigen holders, and they come in two groups of cells. Macrophages can bear both class 1 and class 2 MHC proteins, and then our body cells will also bear class 1 MHC proteins. So let's say that you get infected with just a common cold virus. So your virus gets in, you're up in your nasal mucosa, and the antigen presenting cell finds that cold virus. So it's going to present it on the surface with the class 1 MHC protein, and that's going to activate this CD8 cell. So now we've selected, now we're going to go through clonal expansion, and it's going to make again some memory cells. So these CD8 cells will then wait until further infection by that virus, and then the rest of the cells will then become cytotoxic T cells, the ones that will actually attack the infected body cell. So our infected body cell will have a unit just like this as well, that it will have an antigen held in place by a class 1 MHC protein. So wherever a cytotoxic T cell sees this unit right here, then it will go and cause apoptosis to happen, programmed cell death. So it'll tell your infected mucosal membranes in your nose to carry out apoptosis to reduce the and prevent the spread of the virus. Now, this also needs to be receive chemical signals from the T helper cell, and we'll talk about that in just a minute. All right, now the CD4 cells, on the other hand, are going to get their instruction, their antigen carried on a class 2 MHC protein. And the class 2s are going to be found on a macrophage or on a B cell. And so what the T helper cells are going to do is produce these T cell receptor, well, not produce T cell, they're going to produce their clones, and then again we're going to have memory cells, and then we're going to produce a whole population of either T helper cells or T regulatory cells, and we'll talk about the regulatory cells later. But the T helper cells. So what do the T helper cells do? Well, their job is to command everything. So they're going to send out the chemical signals that are going to activate these cytotoxic T cells in great numbers so that they can fight things off. they're gonna activate the B cells. They will not go through clonal expansion to any great degree without signals that come from the helper T cells. So the helper T cells really are the directors of the battle. So they can be like the lieutenants, or the who's ever in charge, who makes the decisions of the battles and tells everybody what to do. And then you have your people on the front line. You have your non-commissioned, your privates and your corporals and sergeants out there fighting, being told what to do by the T helper cells. Now, here is the link between having AIDS and HIV infection and the T helper cells. So as I mentioned before, there's a receptor called a CCR5 that HIV will bind to in order to find its host cell. Well, the only cells in our bodies that bear CCR5 receptors are the T helper cells. So when we are infected with HIV, that virus will come and bind to and insert its nucleic acid into a helper T cell. And as I told you before, once you've been infected with a virus, you're done. Those cells will lyse. And so what happens when we have HIV infection is the virus will grow inside of the T helper cell. It will lyse the T helper cell. It'll spread to neighboring helper cells, and then proliferate through your entire population of helper cells. Now you have nothing to direct the infection or direct the defense against an infection. So typically individuals with AIDS will die of something that is not something people with healthy immune systems would die of. We'll just take Freddie Mercury, for example. He had AIDS and died of AIDS-related pneumonia. Now, under normal circumstances, he probably could have recovered from that pneumonia, but because there wasn't anything directing his B cells to fight off that pneumonia, then he died. And so that's usually what happens with individuals with HIV infection, is that they will die of something that normally wouldn't kill anybody else with a properly functioning immune system. Okay, so T cells learn self from non-self while in the thymus. T helper cells will stimulate the proliferation of the cytotoxic T cells and the B cells. And notice right here, without CD4 cells to direct it, there's no immune response. Their chemical cytokines will supply the help needed to recruit the other immune cells. So this just shows, here are those chemical signals recruiting the B cells, recruiting the CD8 cells, interleukin 2 trying to and in this case, interleukin 4 in this case, to get the effector cells, so the B cells to become plasma cells, and then to activate the cytotoxic T cells. So that's the role that the helper T cell plays in immunity. And then here's just a bigger video of that. Okay, then cytotoxic T cells will kill 3 groups of cells: virus-infected cells, cancer cells, and tissue transplants. They will secrete a chemical called perforin that will lyse the cell membrane. They'll inject another protein called granzymes will come in and then turn on the apoptosis genes in the infected body cell. T regulatory cells or suppressor cells will inhibit the immune response when the antigens have been destroyed. Typically in an autoimmune disease, we see these T suppressor cells not functioning properly. And so once we've mounted a defense against a particular infection, it's as if our body is looking for something else to kill. And so it just starts attacking our body cells that have an antigen structure that is similar to the antigen that we just killed off. So for example, type 1 diabetes, great example of this. So I had a family of, well, there was just 2 boys, but I work with the parents, and the father has a history of type 1 diabetes in his family. The male members of his family all have type 1 diabetes, but he doesn't. And so both of his sons have become infected, have become type 1 diabetics. And so in the older boy's case, he was in my class, and he had been diagnosed about 4 or 5 years previous to that, maybe not even that long. And so right when he was in my class, his younger brother was diagnosed with type 1 diabetes. And so then we were talking about what the triggers were, and I went and talked to his mom, and I said, so were you able to pinpoint what his trigger was for the younger brother? And she said, oh, I was gonna talk to you. She says, yes, we figured out that hand and mouth disease had been going around through the schools, and that their younger son had contracted that, and it just so happened that the antigens on the hand and mouth virus were similar enough to the antigens on his beta cells within his pancreas that once he got better from the hand and mouth disease, that then hand and foot and mouth disease, that then his type 1 diabetes kicked in. And so then I was talking to the older brother, and he says, well, yeah, we're not really sure because we weren't looking for what my trigger was. They were always checking with the younger brother to see after he got sick, did he have did he come down with type 1? And he says, we think it was a strep throat infection that I had prior to when they first diagnosed him with type 1 diabetes. So there's some autoimmunity lesson, and what happens when T regulatory cells don't work properly is your body just wants to go fight some more things, and there's nobody to tell it no. And that's often why we get an autoimmune disease. Okay, so then T memory cells are going to be the same as B memory cells. Remember the antigen for future infection in the case of future infection, so that you can get that strong and rapid secondary immune response. Here just shows how the cytotoxic T cell is going to bind to the target cell. So this is a cancer cell, virus infected cell, whatever. They'll have the little chemicals in granules, the porphyrins and the granzymes. So the porphyrins are going to come out very similar to complement, poke a little hole, granzymes come in, and then trigger the enzymes that will turn on the genes for apoptosis. This diagram just shows once an antigen enters you, what can happen. So we want the innate defenses to try to keep it at bay, so our surface barriers, our internal defenses, so fever, phagocytes, inflammation, interferon, complement, and inflammation, or our adaptive defenses can just kick in. So here we have either an macrophage engulfing the antigen and then presenting it on its cell surface that then activates either the naive CD4 cells, and then they go through their clonal expansion, and then they send their little chemicals, their co-stimulatory chemicals, cytokines, over here to activate the B cells, or they send their little chemicals over here to activate the CD8 cell. But the APC can activate the CD8 cell itself, but it's not as strong of a response without this co-stimulation by the helper T cells. And they'll go back and go kill things. And then here the B cells, they can present the antigen to the T helper cell, and then those co-stimulation will happen, and then will trigger greater clonal expansion, and then humoral immunity. So this tan side is all cell-mediated immunity, and then the green side is humoral immunity. Okay. So let's really take a look at autoimmune diseases quickly, and then we'll be done with this. So in the case of autoimmune diseases, T cells attack their own antigen. But you know what? With these autoimmunities, it's everybody plays. You've got all the chemical mediators being secreted. You've got antibodies being secreted. You have B cells involved, T cells involved. Just everybody. Macrophages all over the place. I have a poster that has rheumatoid arthritis and MS and type 1 diabetes and one other one, maybe glomerular nephritis. I can't think right now. But it shows everybody that's involved, and there is just a flood of immunity going on in these inappropriate places. So why? Why does it happen? Well, first of all, it may be because we didn't get rid of all the lymphocytes that we should have. We run the risk when the T cells are in the thymus, we run the risk of eliminating T cells that respond to antigens that are similar to our own. Let's say you have, the virus, hand, foot, and mouth virus that looks like the antigens on the surface of this young man's beta cells. That was just by chance that it looked like that. And so our thymus has to decide, do I keep ones that kind of look like self-antigen? That could bind to self-antigens, or should I get rid of everything that could bind to self-antigens? Sometimes we don't. And if we don't get rid of them, then we may keep some around that have the ability to destroy our own body tissues, but may not be activated. Sometimes new self-antigens appear on our bodies, and then they start attacking those new self-antigens. Foreign antigens, again, may resemble self-antigens, and so there's, like with type 1 diabetes, how that happens. Okay, so I'm just going to go through these real quickly. Here's some autoimmune diseases: rheumatoid arthritis. Something caused the inflammatory chemicals to attack the synovial membrane within the joints, and so that begins to be destroyed. In lupus, it's a lot of the connective tissue that is similar to something they've been infected with before, and so the body attacks the connective tissue. MS, I have a niece that has MS, and it's attacking the myelin sheath on the axons of the cells in her body, peripheral neurons primarily. Oh, Graves' disease, that was the other one that I have. This is where the thyroid gland is attacked, and the antibodies will stimulate the production of excessive thyroxine. And then type 1 diabetes, where the beta cells are destroyed, and glomerulonephritis, that will affect the filtration membrane. Okay, I'll just give you these last two things. So congenital immunodeficiency. So this is a you don't have an immune system, you're born without one. Essentially, it's called SCID, or severe combined immunodeficiency. So you don't make enough B and T cells, and so this is the boy in the plastic bubble disease, and so you don't have the T's and B cells to fight off the infection. But it's looking like gene therapy is treating this, and that we are giving them the ability to produce B and T cells, and they're able to fight off infection. So this is the first real genetic congenital disease that we're able to treat with gene therapy. So this is exciting with the treatment of SCID. And then AIDS, of course, is when your CD4 cells, or the T helper cells, are selectively targeted and invaded and destroyed. Okay, so that is all of that.